solution of test 19, and the chapter which is involved in this, that is molecular basis of inheritance. Question 1. Question 1 asks that a complex of ribosome attached to a single strand of RNA. If there is a single strand of RNA and over which a complex of ribosome, means multiple ribosomes are attached. Now this thing is known as polysome or polyribosome or ergasome, and polysome is given in option C. So C is the correct answer. Next is question 2. Question 2 asks that AUG perform two functions. AUG performs two functions. One, it code as a code for methionine, and it act as initiation codon. So it code for methionine and it act as initiation codon. So option B is the correct answer for AUG. Question 3. Question 3 asks that DNA contains. Now deoxy is the deoxyribonucleotide chain. So it is a polynucleotide chain, and nucleotide consists of sugar, then nitrogenous bases and phosphoric acid. So now if we go with the options, option A is given as sulfur cannot be there. Option B is given as phosphorus. Yes, phosphorus is there in phosphoric acid to form a phosphodiester bond. C, boron is not there, and D, calcium, that is also not there. So B is the phosphorus which is used in formation of phosphodiester bond in DNA. Question 4. Question 4 asks that the equivalent of a structural gene is. Equivalent of structural gene is. Now if we talk about the equivalent of structural gene, now functional unit of gene is known as cistron, and it is nothing but a length of DNA that contain information for coding a specific polypeptide. So it has information for the specific polypeptide. Now if we go with the option A is given as muton, and muton is nothing but a segment of DNA which is a unit or segment of DNA where mutation occur. So it is not a structural part. Option B is given as cistron. So yes, cistron can be possible. Option C is given as operon. Operon is a group of genes, so it cannot be a structural unit. And D is the recon. Recon is a unit of recombination. Muton is a unit of mutation. So cistron is the correct answer, that is option B. Question 5. Question 5 asks that the unequivocal proof of that DNA as the genetic material. So DNA unequivocal proof given by Alfred Hershey and Martha Chase experiment, and this experiment was carried out in 1952, and by use of a bacteriophage. And bacteriophage is nothing but a virus or bacterial virus which infect bacteria. So D is the correct answer. Question 6. Question 6 asks that which of the following is false for RNA? If we go with the option 1 is given as RNA can directly code for synthesis of protein. So yes, RNA directly code for protein. Second is given as it is very common process to synthesize DNA from RNA. DNA from RNA, then it is known as reverse transcription, and reverse transcription is not common as it is found in few viruses only which has a RNA as a genetic material. 3. RNA is used as genetic material only in human. It is incorrect. RNA is not used as genetic material in human. And option 4, one polypeptide chain is present in RNA. Yes, in RNA single polynucleotide chain is found, so it is correct. So incorrect are or false are second and third. So second and third is incorrect. So option C is the correct answer. Question 7. Question 7 asks that it is an expression of Y gene in lycopyron. Now in lycopyron there are three genes, three structure gene: leg Z, leg Y and leg A. Leg Z code for the beta galactosidase, leg Y code for a permease, and leg A code for the transacetylase. Now the question is the expression of Y gene in lycopyron. Now leg Y gene code for the permease, so permease is given in option B, so B is the correct answer. Question 8. Question 8 asks that in a DNA molecule, option A, the proportion of adenine in relation to thymine varies with the organism. Now proportion of adenine is equal to thymine. It does not vary with the individual base because a number of adenine is equal to number of thymine, so it is incorrect. Option B is given as there are two strands which run in anti-parallel. One is in 5' to 3' and another one is in 3' to 5', so it is correct. C is given as the total amount of purine nucleotide and pyrimidine nucleotide are always equal. Purine nucleotide and pyrimidine nucleotide is always equal. Now purine are adenine and guanine, and pyrimidine cytosine and thymine. So now this adenine pair with thymine and guanine pair with cytosine, so always the number of adenine and guanine is equal to number of cytosine and cytosine. The purine is equal to pyrimidine, and statement is not always equal, so it is incorrect. D is given as there are two strands with one with run parallel in the 5' to 3' direction. It is incorrect, that they run in anti-parallel nature as option in the option B, so B is the correct answer. Question 9. Question 9 asks that in the lycopyron, which one is responsible for code of repressor? So repressor is an inhibitor that is coded by a regulatory gene, and regulatory gene This is the I gene. So option A is the correct answer. Question 10. Question 10 asks that spliceosomes are not found in the cells of. Spliceosomes are not found in cells of. Now spliceosomes are found during the splicing, and splicing occur when there is a presence of intron in DNA sequence. Now eukaryote have intron in their DNA sequence. Bacteria lack intron sequence. So spliceosome is not required, or splicing is not required in bacteria. So C, bacteria is the correct answer. Question 11. Question 11 asks that which option is matched with the figure. So the figure, according to NCERT, it represent option A, that is polyadenylation, and polyadenylation is also known as tailing. So C, A, B, both are true. That is the correct answer. D is none of these, so it is not a correct answer. Question 12. Question 12 asks that which one of the following does not follow central dogma of molecular biology. It does not follow central dogma. Now central dogma is a one-way flow of information. It is a one-way flow of information from DNA to mRNA and mRNA to protein. Now this is found in case of particularly all organism, like option A, P, yes, it is found, it is eukaryote. B, mucor, yes, it is a yeast. C, in chlamydomonas, yes. In D, HIV. Now in HIV, RNA is genetic material. Now the organism in which RNA is genetic material, DNA get also synthesized. So the organism in which RNA is genetic material, like HIV, reverse transcription occur. That is formation of DNA from the RNA. So central dogma is not found in case of HIV. Question 13. Question 13 asks that formation of RNA strand on template DNA is. Now formation of RNA on template DNA, that is formation of RNA from the template DNA. This process is known as transcription. So B is the correct answer. Question 14. Question 14 asks that amino acid sequence in protein synthesis is decided by the amino acid sequence in protein decided by genetic code present on mRNA. So mRNA, C is the correct answer, and this mRNA have a genetic code in the form of triplet code that is responsible for defining the amino acid sequence on the protein. So mRNA is the correct answer. Question 15. Now question 15 asks that a molecule that act as genetic material that must fulfill the traits given below except. Now A, it should be able to express itself in the form of Mendelian character. Yes, the genetic material is suppose DNA, then it should be able to express in the form of what? A Mendelian character. Option B, it should be able to generate its replica. Yes, replication, it should be able to carry out replication. So it should be unstable structurally and chemically. It should be stable structurally and chemically. Should not be unstable, but his statement unstable is given, so C is incorrect. And D is given that it should provide a scope for slow changes that are required for evolution. Yes, slow changes should be available, so mutation can occur, and that is responsible for the evolution. So C is the correct answer, that it should be stable.